In other news, the inaugural U.S.-Saudi Biotech Alliance Summit will commence today from San Francisco. And leading up to the event, ImmunityBio announced that it's received positive results from its Anktiva clinical program in non-small cell lung cancer based off of a pair of studies. J.D. Durkin spoke with Dr. Patrick Shiong, executive chairman at ImmunityBio and Networks and owner of LA Times Media Group, about the key breakthroughs from these results. Let's take a look.
Well, the key breakthrough was the fact that forever now, with patients who received checkpoint inhibitors, received chemotherapy called docetaxel and had about a 7 to 9 months survival. We've now shown with this combination of checkpoints plus Anktiva, we've almost tripled that survival. So that was the important result that we showed today, was that we can increase the survival by maintaining your immune competence.
Checkpoint inhibitors work, doctor, until they don't. What is failing in the immune system whenever patients relapse?
So as you know, in the market, both Merck, Pfizer, Bristol Myers have had multiple checkpoint inhibitors, and it's been a very big and major product. And the way checkpoint inhibitors work is by taking the brakes off T cells. The problem is that's an event that happens temporarily because the T cells get exhausted. Once the T cells get exhausted, unfortunately, the patients recur. So while checkpoint has been a really important addition now to the armamentarium of starting immunotherapy, it is not the full answer, because patients have a very temporary response to that and finally get progression.
Dr. Shiong, why is absolute lymphocyte count such a critical signal, and why has it been overlooked?
So the absolute lymphocyte count is the number of cells in your body that are important to kill cancer. And these cells are the T cells that is activated by the checkpoint, but they have to be talking to other cells called the natural killer cells. So the absolute lymphocyte count is a measure of both all these cells that are actually responsible for killing cancer. It's been there for years and years. In fact, there's a reimbursable code to be able to measure it from your CBC, and nobody's used that because, unfortunately, until today, there's never been a treatment to reverse this lymphopenia. So if one were to Google lymphopenia and look at mortality rates, you will see, even in the US general population, if you have a low ALC count, you have a lower survival rate. If you have cancer, you have a low ALC count, regardless of the cancer type, you have a lower survival rate. If you have sepsis, you have a lower survival rate. So the idea then that this ALC count matters, but the difference is today we can now change and modify and improve that ALC count. So no longer is it just a diagnostic test, it's a modifiable, treatable of an indication of whatever disease is by improving the ALC and thereby improving your immune system.
Let's drill down a little bit more about the underlying data here, Dr. Shiong. Your data links immune restoration directly to longer survival. How strong is that connection?
Well, what was exciting, we unblinded a randomized trial. And by a randomized trial, we were able to prove that when you add Anktiva, you increase the ALC count. So the next step was to analyze patients who had failed all treatment, even checkpoint, and while progressing on the checkpoint, and all you did is add Anktiva, and show that when you maintain and increase the ALC count, does that change survival? And the answer is statistically significantly so, that we achieved up to 21 months of what they call median survival. We have patients now going over 3 years, and we will show a video of these patients that were on these trials, receiving merely now Anktiva plus a checkpoint inhibitor, now having much prolonged median overall survival. But the most important thing is to have the survival with a great quality of life, because the chemotherapy currently, the standard of care, docetaxel, is highly toxic.
You've called this immunotherapy 2.0. In simple terms, how is this different from today's standard approach?
Well, today's standard approach is exactly what it is checkpoint inhibitors. But now, for the first time, the checkpoint inhibitors can be supplemented with natural killer cells, which sort of show you have T plus NK cells. Now that becomes, I consider, really the foundational platform for what I call immunotherapy 2.0, because it's a complex system that's going on in your body. The tumor is trying to hide from the immune system, the tumor is trying to block your immune system, and your immune system is getting tired. If we can now activate the immune system with NK cells and T cells, and more importantly, add to the patient an infusion of natural killer cells that we've actually grown in the laboratory under GMP conditions, you begin to see a complete new platform and a paradigm change of immunotherapy 2.0.